At Lotus, we have spent years honing our clinical trial skills so that we can produce the greatest standardized effect size possible. We do this because the standardized effect size, or SES, is the key variable that determines the result of a clinical investigation. The inherent efficacy of the study molecule is only one factor that contributes to the probability of a positive clinical trial. There are many things that the site and the sponsor can do collaboratively to optimize the clinical trial and increase the probability of success. In the following video, we will describe why the SES is so important and what we at Lotus do to maximize it. In order for any clinical trial to be successful, active medication must separate from placebo without significant variability. In a typical analgesic investigation, patients are randomized into the clinical trial with a baseline pain intensity of approximately 5. Patients randomized to placebo should have pain scores that worsen or improve minimally, while patients randomized to an effective study medication should significantly improve. The space between is the observed difference. Each arm has some variability, and the resultant green area graphically represents the SES. We want to maximize the SES in order to make the probability of a positive investigation as great as possible. Many analgesic studies are negative even though the investigational product has efficacy because the SES is artificially lowered by experimental error. In an analgesic investigation, the SES has 3 primary enemies: high placebo response rates, suboptimal treatment effect from the investigational medication, and a high degree of variability, also known as a large standard deviation. The formula for SES is drug effect minus placebo effect divided by variability. The SES is not fixed. It will vary based on the quality of the experiment. If study power and confidence intervals are held constant, the N required for a positive clinical investigation is inversely proportional to the square of the standardized effect size. To demonstrate the point, we examine the SES and required N for a known analgesic agent, IV morphine. IV morphine has reliable efficacy, but some investigations in the literature show an SES below 0.3 and others demonstrate an SES greater than 0.6. One can see that doubling the SES has a drastic impact on the required N. Armed with all of this information, we at Lotus take great pains to examine and consistently improve the SES generated from our analgesic investigations. At the end of every trial, we calculate the Lotus SES and compare it to the SES generated at non-Lotus sites so we can understand how to improve the quality of our investigations. Utilizing our knowledge and techniques, experimental error can be reduced. Treatment effect can be maximized by picking the appropriate patient population for the study agent, giving the study agent at the appropriate time in the disease course, and finding patients who will most likely respond to the investigational product. Placebo response is an issue that is tightly tied to the management and conduct of the study at the site level. A properly educated patient is your greatest ally in reducing the placebo response. Specifically, patients need to understand that they are in a research alliance and not in a therapeutic relationship. We have videos to educate the patient, some of which can be found on our website. Another key factor is educating study coordinators so that questionnaires can be administered without bias. The last component is variability reduction, which can be achieved by using as few centers as possible in order to meet enrollment goals, and standardizing subjective portions of the protocol amongst the various study centers and study coordinators that will be performing the trial. At Lotus, examining, understanding, and improving our SES in analgesic studies is our core mission. We have a significant amount of data that indicates our SES is better than that generated at most other analgesic sites. In many cases, we have seen that our resultant SES is 50 to 90% higher than the norm. This translates into a need for less patients and a higher probability of clinical trial success. See our website for our methodology publications, which describe past clinical trials and the SES numbers that we achieved in various models. Feel free to contact us to discuss SES maximization.